Good morning, everyone. Continuing our discussion on transgenic plants. In today's session, we will see the methods for the development of transgenic plants. The transfer of exogenous DNA to higher plants can be accomplished by various methods. So, there are various methods available to transfer a foreign DNA or exogenous DNA to higher plants. In today's session, we will focus only the classical and widely used methodologies for the purpose of developing a transgenic plant. So our discussion will be on the classical and the most widely used methodologies for the development of transgenic plants. To begin with, we have the genetic transformation using Agrobacterium tumefaciens. We all know that the Agrobacterium tumefaciens is an aerobic gram-negative bacterial species that belongs to the Rhizobiaceae family, and it is found in soil. It is a pathogen that infects a wide range of hosts of plants. So that is about, or it is more than 600 species. This particular gram-negative aerobic bacteria has the capability to infect more than 600 species of the plants. And this bacterium causes crown gall, which is a tumor forming disease in the infected plants, and it has the ability to transfer a part of its genetic material into the host plant, and the consequence is there is alteration in the gene expression of the host for its own benefit. So this particular gram-negative bacteria has the ability to transfer part of its genetic material into the host plant, thus altering the gene expression of the host that makes suitable for the growth and development of this Agrobacterium tumefaciens. So there are related species, say for example Agrobacterium rhizogenes. This causes the proliferation of secondary roots at the point of infection, and the Agrobacterium rhizogenes is also explored extensively to be used to transform the plants. So we have Agrobacterium tumefaciens, which infects wide range of host plants, over 600 species. And there are several related species. To give an example, the Agrobacterium rhizogenes, which causes the proliferation of secondary roots at the point of infection, is also explored to be used for transforming the plant. Right. So in the initial stages, that means during the initial stages of the Agrobacterium tumefaciens infection process, the bacteria will recognize and contact the plant cell. So two things happen. One, the recognition, and number two, contact development. So this happens in the initial stages of the Agrobacterium tumefaciens infection process. So the bacterium recognize and contact the plant cell. Now this process of recognition and contact, it is mediated by signaling molecules such as low molecular weight phenolic compounds or amino acids and or sugars released by the infected or injured root. Right. So the process of recognition and contact with the plant cell is mediated by signaling molecules. These molecules act as signals, and these molecules are, for example, low molecular weight phenolic compounds, amino acids, and sugars released by the injured root. So after the contact is established, the signaling molecules, they promote the activation of genes, specifically the virulence genes. The virulence genes, which is abbreviated as VIR genes. Now these are located in the VIR region, the VIR region, where of the tumor inducing, that is TI plasmid in Agrobacterium tumefaciens. Right. So carefully understand the process. So during the initial stage of Agrobacterium tumefaciens infection process, the bacterium will recognize and contact the plant cell. Now this process of recognition and contact of the plant cell, it is mediated by signaling molecules such as phenolic compounds, amino acids and or sugars which are released by the injured roots. Now once the contact is established, what happens? The signaling molecules, they promote the activation of virulence genes, the VIR genes, which are located in the VIR region or VIR region of the tumor inducing, that is TI plasmid in Agrobacterium tumefaciens. Right. Now, further, there are 6 groups of genes in the VIR region that are either essential. So the essential ones are VIRA gene, VIRB gene, VIRC and VIRD gene. That is VIR-A gene, VIR-B gene, VIR-C gene and VIR-D gene. So these are the essential genes, and 2 more are there for the function to increase the efficiency of the host cell's transformation, that is VIR-E gene and VIR-G gene. Right. So there are 6 groups of genes in the VIR region, and additionally you find other virulence genes located on the bacterial chromosomes, and they are also involved in the early recognition and contact stages of the infection process apart from these 6 groups of genes. Right. So there are VIR-A, B, C, D, E and G genes. Additionally, other virulence genes located on the bacterial chromosomes, they are also found to be involved in the early recognition and contact stages of the infection process. Right. So the proteins which are encoded by these VIR genes, they are most essential for the transfer of the tDNA, that is transferred DNA. that is tDNA region of the Agrobacterium tumefaciens Ti plasmid to the nucleus of the host plant cell. Right. So these genes, that is the product of these genes, the proteins encoded by Vir genes, they are essential for the transfer of the tDNA region of the Agrobacterium tumefaciens Ti plasmid to the nucleus of the host plant cell. Right. Now what happens? The tDNA region is flanked by left and right border segments. Now these are repeated sequences of approximately 25 base pairs. Right. Now the left and right borders, they are most critical for the recognition of the transfer region that must be released. Now, a single strand of the tDNA complex is transferred along with the Vir genes to the plant cell nucleus where it is inserted and expressed. Right? Now, the tDNA which is transferred into the nucleus of the host plant cell, this tDNA region is flanked by left and right border segments, so which are repeated sequences of approximately 25 base pairs. Now the left and right borders are critical for the recognition of the transfer region that must be released. Right? So a single strand of the transfer DNA complex is transferred along with the Vir genes to the plant cell nucleus where it is inserted and expressed. So I am essentially trying to make you understand the process of the transfer of the tDNA complex and its insertion and expression. So it is believed now that its integration into the genome of the plant cell is random. But there are indications that integration occurs specifically in transcriptionally active regions. So there are indications, but by and large, it is believed that the integration into the genome is random, but there are some reports which indicate that integration occurs specifically in transcriptionally active regions. Right? Now, if you just look at this illustration, this is about a schematic representation of the Ti plasmid of Agrobacterium tumefaciens and its modifications for plant transformation. You have four figures over here, figure A, B, C and D. So the A represents the Ti plasmid, B represent the co-integrated plasmid, C represent helper Ti plasmid and the D represent binary vector. So it is a schematic representation of the Ti plasmid of Agrobacterium tumefaciens and its modifications for plant transformation. So what happens in A, B, C and D? What is the information that we are trying to obtain? In A, the Agrobacterium tumefaciens native Ti plasmid. So what is depicted in A is, it is the Agrobacterium tumefaciens native Ti plasmid. So the oncogenes responsible for the production of the cytokine, that is abbreviated as PC over there, and auxin, it is represented by PA, as well as the gene responsible for opine synthesis represented as SO. If you carefully look at the plasmid in the figure A, you have PC, which is for the production of cytokinin, and you have the PA, which is referred for auxin, as well as you have the SO referred to for opine synthesis, and these are found in the transfer DNA, that is tDNA region. Right. So the oncogenes responsible for the production of cytokinin, auxin, as well as the gene responsible for opine synthesis, they are found in the tDNA region. Now the tDNA region is flanked by the right border, that is RB, and the left border depicted as LB. So the right border and left border regions, which are both essential for the tDNA transfer process. So if we carefully look at the illustration over there, LB and RB are shown over in figure A, which is the right border and the left border regions, which are both essential for the tDNA transfer process. So in addition, the Ti plasmid has a virulence region, that is Vir region, a region responsible for conjugative transfer, that is DC, which is given in the green code, the TC, that is referred to as conjugative transfer. That is Ti plasmid has a virulence region, that is Vir region, a region responsible for conjugative transfer, that is TC. A TC, it is a region responsible for expression of genes involved in opine catabolism, that is referred to as CO, and the origin of replication for Agrobacterium tumefaciens, that is OR. So in addition to PA, PC, SO and the left border LB and the right border RB, the Ti plasmid has virulence region, and it has a region for responsible for conjugative transfer, that is TC, and it is also having a region responsible for expression of genes involved in opine catabolism, that is CO, and finally it has a region for the origin of replication for Agrobacterium tumefaciens, that is OR. So this is essentially about the Ti plasmid structure. Now, if we just move on to the figure B, to disarm the Ti plasmid, the PC, that is for cytokinin, the PA, that is for auxin, the SO for opine synthesis, the TC for conjugative transfer, and the CO for opine catabolism. All these regions of the tDNA are removed and an exogenous tDNA section is inserted. So where exactly the insert find its place? So to disarm the Ti plasmid, the PC, PA, SO, TC and CO regions of the tDNA are removed and an exogenous tDNA section is inserted. Right. Now the exogenous tDNA introduces the exogenous gene of interest, which is referred to as GI, and the selection marker gene referred to as SMG, and the promoter P and the terminator T regions. Right. So to disarm the Ti plasmid, you have to remove PC, PA, SO, TC and CO regions of the tDNA and an exogenous tDNA section is inserted. Now the exogenous tDNA introduces the exogenous gene of interest, the selection marker gene, the SMG and the promoter P and terminator T regions. Now additionally, an antibiotic resistance gene, that is referred to as R, is inserted to allow for selection of transformed bacteria. Right? So this is about the disarmed tDNA process. Now, if you just carefully move on to the figure C, the Ti plasmid with excised or removed tDNA, it is used as a helper in the tDNA binary vector system. Right? So the tDNA plasmid with excised tDNA or removed tDNA is used. That means complete tDNA is removed. Then this Ti plasmid is used as a helper in the tDNA binary vector system. Now, if we just move on to the figure D, this is about a binary vector has the gene of interest as well as the selection marker genes for both Agrobacterium tumefaciens, that is SMG2 represented over there, and the plants, that is SMG1. One is represented in green color, the other one is represented in yellow color. So the binary vector has the gene of interest as well as the selection marker genes for both Agrobacterium tumefaciens as well as for the plants. And it also has an origin of replication for E. coli, that is ORE, small e, and it carries a helper Ti plasmid containing the binary vector which can be used for plant transformation. Right. So this is about the Ti plasmid of Agrobacterium tumefaciens and its modifications for plant transformations. Because the Ti plasmid is very large, say for example approximately 2 lakh base pairs, it is very much necessary to use smaller sized plasmids. So they are easy to handle, right, and very well capable enough for the process of transformation. So essentially these vectors should contain the tDNA flanking regions and they may be integrated into a disarmed Ti plasmid to produce a co-integrated plasmid, or simply it be a binary vector which is independent of a Ti plasmid. Is that clear? So essentially we have tried to understand about the Ti plasmid, the co-integrated plasmid, the helper Ti plasmid and the binary vector system. Right? So the binary vectors, they are maintained separately from Ti plasmids and they are derived from plasmids that can or that have the ability to replicate in both E. coli and Agrobacterium tumefaciens. And the co-integrated vectors, they result from the integration of intermediary vectors that do not replicate in Agrobacterium tumefaciens with the Ti plasmid. So this which occurs via simple recombination using a region homologous to the disarmed strain. So essentially we are trying to understand the difference between a binary vector system and a co-integrated vector system with reference to Agrobacterium tumefaciens. So the binary vectors are maintained separately from Ti plasmids and they are derived from plasmids that can replicate in both E. coli and Agrobacterium tumefaciens. The co-integrated vectors on the other hand result from the integration of intermediary vectors that do not replicate in Agrobacterium tumefaciens with the Ti plasmid, which occurs via simple recombination using a region homologous to the disarmed strain. Is that clear? Okay. Now, there are three common methods that are used for vector transfer. Number one, the triparental conjugation. Number two, electroporation. And number three, the heat shock. So, the three common methods which are used for vector transfer are: number one, triparental conjugation. electroporation, and heat shock. Now, the choice of method depends primarily on the resources available in the given laboratory. Because all the three methods given over here used for vector transfer, they are equally effective, right? So the methods are triparental conjugation, electroporation, and heat shock. So all the methods are equally effective, and the choice of the method that depends upon the or primarily on the resources available in the laboratory. The triparental conjugation, it is based on the use of donor, recipient, and helper bacteria. Now, for this method, two strains of E. coli, that is the donor and the helper, and a strain of a recipient, where in this case it is Agrobacterium tumefaciens, they are cultured together. Right? So for this method, two strains of E. coli, the donor and the helper, and a strain of a recipient, that is Agrobacterium tumefaciens, they are cultured together. Now the helper E. coli promotes the mobilization and transfer of the plasmids between compatible bacteria. Now this process is mediated by mobilization, that is MOB genes, and transfer, that is TRA genes in the helper plasmid. So the donor E. coli carries the binary vector that will be transferred to the recipient bacterium, and during the co-cultivation of these bacteria, the helper plasmid is transferred to the donor E. coli. Right? So if you just carefully go through this illustration, there are two steps, that is step 1 and step 2. Step 1 is about conjugation initiated by the self-mobilizable helper plasmid, and step 2 is about the donor plasmid. Now, subsequently, this plasmid now present in the donor bacterium promotes the mobilization of both itself and the binary vector into Agrobacterium tumefaciens. The helper plasmid is unable to replicate in Agrobacterium tumefaciens and is simultaneously eliminated. So the helper plasmid which are unable to replicate in Agrobacterium tumefaciens, they are spontaneously eliminated. The strain of recombinant Agrobacterium tumefaciens, now it is selected as usual with the appropriate antibiotics. Is that clear? So the helper plasmid which are unable to replicate in Agrobacterium tumefaciens, they are spontaneously eliminated, and the strain of recombinant Agrobacterium tumefaciens is then selected as usual with the appropriate antibiotics. Right? And next we have the electroporation. What happens in electroporation? Electroporation, in this case, the vector, that is binary or co-integrated, and Agrobacterium tumefaciens competent cells are together subjected to a higher voltage electric pulse. So the vector and the bacterium competent cells are together subjected to a higher voltage electrical pulse. Right. Now the generated electric field, it promotes a reversible destabilization of the plasma membrane, thus allowing the vector to enter the Agrobacterium tumefaciens so that the tDNA is transfer, right? And next we have the heat shock method. In the heat shock method, the permeability of the Agrobacterium tumefaciens plasma membrane is reversibly changed by successive incubation at extreme temperatures. So starting from minus 186 degree centigrade to 37 degree centigrade. And these changes to the plasma membrane allow the vector into the Agrobacterium tumefaciens cell, right? So these are the three important methods which are about the transfer of the vector into Agrobacterium tumefaciens cell. So I'll stop my discussion over here. We can continue our discussion in the next session. Thank you very much.